So students, now we will talk about histology of liver. And liver is the biggest gland in our body and biggest digestive gland, and it provides plenty of functions. So functions of liver are the following. It provides bile synthesis and secretion. It provides emulsification. Bile is released in the digestive tract, digestive tube, and bile acids, they provide emulsification of fats. And fats after emulsification are accessible for lipase, which is produced by the pancreas. So bile, it participates in the digestion of fats. And also its excretion of bilirubin, excretory function. Bilirubin is a product of metabolism of hemoglobin, and it's released by liver. Also, liver provides protein synthesis, especially proteins of blood plasma. It provides gluconeogenesis, production of glucose. It's storage of different substances such as proteins, such as vitamins A, D, E, K. Also, it's storage of glycogen and lipids. So it keeps them and it accumulates them for all needs of organism. Liver is involved in detoxification. Many different medicines, for example, drugs, different um, toxins, they are going to liver, and liver makes them not dangerous. It's called detoxification. Also, liver provides protective function, and also liver is hemopoietic organ during embryonic period, and also liver provides endocrine function. Some endocrine regulative substances are produced by the liver and released in the bloodstream. And let's begin from anatomy and microanatomy of liver. So liver is divided into lobes. We have 4 lobes, and lobes of liver, they are divided into segments. Here we can see segments of liver, but segments are also divided into smaller units, and the structural and functional unit of liver is lobule. So lobule is the structural and functional unit of liver. Here we can see a histological slide. It's liver of pig, and pig's liver, it has well-developed septa of connective tissue. Here we can see septa of connective tissue, which also divide this gland into lobules. Those lobules particularly remind us of lobules which are found in salivary glands, in pancreas. So there are septa which divide the gland into the lobules. And here we can see lobule. Lobules of liver are hexagonal in shape. Here we can see hexagon, and there is central vein in the center. Here we can see shape of the hepatic lobule. It's hexagonal in shape, and inside we have central vein and neighboring lobules. And here we can see this pattern of localization of hepatic lobules. And between them we have connective tissue. There are septa or interlobular connective tissue layers. And let's look at the shape of classical hepatic lobule. On two-dimensional slides, we can see that lobule has shape of hexagon, and in three-dimensional images, we can see that classical hepatic lobule is hexagonal prism. Here we can see this prism shape of the hepatic lobule. And here we can find central vein. It's located in the center of the classical hepatic lobule. And those lobules are arranged in this pattern. They are attached to each other. In human liver, we cannot find clear borders between the hepatic lobules. Those septa, they are well developed in pig's liver, but in human liver, we can see that there are no clear borders between lobules, but we can find here hexagonal region which surrounds central vein and hexagonal region around this vein, and its lobule in human liver. If human liver has well-developed septa between the lobules, It's pathological condition. It's fibrosis which may develop into cirrhosis. It's very difficult, very severe disease of liver. So here we can see hepatic lobule in human liver. And hepatic lobule has at its center its central vein, and also there are hepatic plates. There are cords of cells. They are called hepatocytes. Main cells of liver are called hepatocytes. They are located radially, and between them there are sinusoids, modified capillaries which contain blood, and also they are located radially. Let's look here. Here we can see a central vein, and there are cords of cells, rows of cells, and they are located radially, like rays of sun, and between them empty or almost empty places, white regions, are sinusoids, modified capillaries, which are going also to the central vein. So we can see radial localization of the structural components of hepatic lobule. And main hepatic cell is hepatocyte. They are classical cells which have all essential organelles. They have full set of general organelles, including rough endoplasmic reticulum, smooth endoplasmic reticulum, Golgi complex, also mitochondria, lysosomes, peroxisomes, and also they have inclusions of fat, of glucagon. And here we can see shape of hepatocyte. It has a shape which is close to cube, so cuboid shape. It's cube-like cell, and it has 6 surfaces. And we will use this cube model to construct hepatic lobule. Let's look how hepatic lobule and hepatic plate is made up of hepatocytes. So there are hepatic plates, rows of hepatic cells, which are going to the central vein, are called hepatic plates, and they are composed of hepatocytes. Here we can see one and second hepatocytes, and they are attached to each other via intercellular junctions. Here we can see intercellular junctions, and they attach neighboring cells to each other. And between the cells we have gaps, special spaces, gaps, and those gaps, they are filled with some fluid, and this fluid is bile. So there is gap between two cells which is filled with bile, and this space with bile is called bile canaliculi, or bile capillaries, and they are located between their neighboring hepatocytes. Bile canaliculi, they don't have own wall. Their wall is formed by cytolemma of hepatocytes. So here we can see cell membrane of one hepatocyte and cell membrane of another hepatocyte, and they form the wall of bile capillary or bile canaliculus. And there are microvilli of hepatocytes which form this wavy contour of bile capillary. It's because of presence of microvilli. They increase surface of hepatocyte, which is involved in the formation of bile canaliculus. And here we can see pair of hepatocytes. It's bile capillary, and there are rows of hepatocytes, and here we can see following pairs of hepatocytes, and we can find that this bile capillary goes through all hepatic plate. So we can see here hepatic plate. It contains 2 rows of hepatocytes, and inside this hepatic plate we have bile capillary, which goes through all hepatic plate. And if we will cut this hepatic plate in this plane, we will see this picture. We will see hepatic capillary, bile capillary or bile canaliculus, which goes through all hepatic plate. Here we can see this bile canaliculus or bile capillary, and it's filled with bile. This wavy contour is result of presence of microvilli on hepatocytes. They have microvilli which enter in the bile canaliculi. But bile canaliculi, they don't have own wall because their wall is formed by hepatocytes. Hepatocytes, their cell membranes, they form wall of bile canaliculi. And here we can see hepatic plate, its view of the outer surface and its view on the section. And here we can see two layers or two rows of hepatocytes, and between them we have bile capillary or bile canaliculi. Here you can see hepatic plate. And hepatic plate is composed of hepatocytes. It's two cells thick, and on another sections it may be one cell thick if section goes in this direction, in this plane. And bile canaliculi wall is made of hepatocytes. So here we can see this hepatic plate. And here we can see two neighboring hepatocytes. It's a cell membrane of one and it's cell membrane of another hepatocyte, and between them we can see the gap. It's filled with bile. It's bile canaliculi. There are microvilli of one hepatocyte and microvilli of another hepatocyte. And here we can see hepatocytes. There are hepatic plates, and between them there is bile canaliculi. And also there are hepatic plates. They are going radially to the central vein, and between them we have blood vessels. Those blood vessels are called sinusoids. Here we can see hepatocytes. It's bile canaliculi, gap between two hepatocytes filled with bile. And here we can see hepatic plate and its bile canaliculi, and there are hepatocytes. And let's look how the hepatic lobule is formed by hepatic plate. So here we have hepatic plate. It's composed of hepatocytes and its hepatic plate, and gap filled with bile, it's bile canaliculi. And hepatic plates, they have neighboring structures which surround it. There are blood capillaries which have huge gaps between endothelial cells, and those capillaries are called sinusoids. They are capillaries of sinusoid type. And cells of the wall of sinusoid are called endotheliocytes. It's simple squamous epithelium. Also, we have here neighboring hepatic plates. So here we can see the pattern: plate, sinusoid, hepatic plate, sinusoid, and following hepatic plate. And inside each hepatic plate we have bile canaliculi. So hepatocytes, sinusoid, also layer of hepatocytes, bile canaliculi, next layer of hepatocytes, sinusoids, and next hepatocytes. So here we can see this pattern. And also here we can see that sinusoids, they are opened in the central vein. Here we can see central vein, and it's lined with endothelium, simple squamous epithelium. And under the endothelium we have basal membrane. Here we can see, and it's incomplete. It's present near the central vein, near the central part of the hepatic lobule, and in the peripheral part. But central part of the hepatic plate, it doesn't have basal membrane, and in normal condition it's absent here. And between the hepatic cells, between hepatocytes and endothelium of sinusoid, we have space which is called perisinusoid space of Disse, or space of Disse. It's filled with plasma of blood. Blood is circulating here in sinusoid, and this blood plasma, it enters this space of Disse. It's distributed here, and some substances from this plasma are uptaken by hepatocytes. And let's look how it's working together. So here we can see blood. Blood is circulating here in the sinusoids, and it goes from the periphery of hepatic lobule to the center and central vein. it collects blood from all sinusoids, and they are radially oriented to the central vein. So it is collector of all blood of hepatic lobule, and blood of sinusoids goes to the central vein. And this blood, it may contain different harmful substances or different metabolites or different drugs, and something comes here to the liver, some substance goes here, and it goes to the space of Disse, it passes through the endothelium, goes to space of Disse, and goes to hepatocyte. And this surface of hepatocyte, which is oriented to the blood vessel, is called vascular pole or vascular surface of hepatocyte. It's located near the vessel. Hepatocyte uptakes this substance and it provides processing of this substance. It provides detoxification or further metabolism. So here we can see metabolism of this substance. And after that, this substance is neutralized, for example, detoxified, and after that it's released into bile. Here we can see release into bile, into the bile capillary, and it's way how bile is produced. So hepatocytes, they produce bile from the components which they uptake from blood. And this pole of the hepatocyte or the surface hepatocyte which releases different substances is called biliary pole or biliary surface of hepatocyte. And this substance goes from the center of hepatic lobule to the periphery in the opposite direction with blood. Blood goes from the periphery to the center and bile from the center to periphery. And here we can see that blood and bile, they are moving in opposite directions. And after the production of bile in the hepatic plate, it fills bile canaliculi and goes from the center to periphery of the hepatic plate. And what is the further way of the bile? There is small duct or duct-like structure. It's called cholangiole. It's like intercalated duct in the salivary glands or in the pancreas. It's called cholangiole. And cholangiole is opened into the bigger duct. It's bile duct, which is located on the margin of the hepatic lobule. It's lined usually with simple cuboid epithelium. Also, it has basal membrane. It has layer of connective tissue, with fibrous connective tissue, and it's called bile ductile or bile duct. And those bile ducts, they form network. Smaller bile ducts, they form bigger, and they form common bile ducts of the lobes. And after that, they form common hepatic duct. So bile from bile canaliculi goes to cholangiole and to bile duct, which carries this bile and forms network of bile ducts inside the liver. So here you can see bile outflow, bile circulation in the liver. And hepatic sinusoid, special capillary, special vessel, which is located between the hepatic plates. It's lined with endothelium. It has basal membrane with gaps. It's present in the beginning and near the end of hepatic plate. And also there are hepatic macrophages or stellate macrophages, stellate cells. They are called Kupffer cells. Here we can see Kupffer cell, and this Kupffer cell, it's immune cell, which provides immune functions of liver. It takes different harmful substances, this Kupffer cell, and it's located between the endothelial cells or it's located above them. And sometimes it's fixed and sometimes it might be movable. And between the endothelial cells and hepatocytes, there is space of Disse or perisinusoidal space of Disse. Here we can see structure of hepatic plates. Here we can see two rows of cells. It's hepatic plate. Between those hepatocytes, we have bile canaliculi. and spaces filled with blood, they are sinusoids, which are opened into the central vein. Here we can see bile canaliculi between the hepatocytes, its hepatic plate, and we have here sinusoid, its Kupffer cell, and its base of sinusoid. And let's look what else cells are present in liver. So the most important cells of liver, the most known cells are hepatocytes. Also, there are neighboring hepatocytes. They form hepatic plates. Between them, there is a blood capillary or bile capillary or bile canaliculi. And also we have here wall of the sinusoid. There are endothelial cells or endotheliosides. Inside the wall, between the endotheliosides, there are Kupffer cells. It's macrophage of liver. It's Kupffer cell or stellate macrophage. Between the wall of sinusoid and hepatocytes, we have perisinusoidal space of Disse. It's lumen of sinusoid, which is filled with blood, and it's perisinusoidal space of Disse. And in this space between endothelial cells and hepatocytes, we have special cells. Here we can see this cell. It has triangular shape. It has processes which enter between hepatic cells and which enter in the space of Disse. And those cells are called itos cells. This itos cell or perisinusoidal lipocyte, it has granules which contains vitamins A, D, E, K. They are dissolved in their fats. So here we can see granules with those vitamins, and also itos cells, they produce reticular fibers and collagen fibers. So here we can see those cells. And fourth type of hepatic cells, it's pit cell. Here we can see the cell. It's very close in functions and structure to the natural killers type of lymphocytes. They are present in liver. And some scientists describe pit cells as a kind of natural killers. So here we can see main cells of liver. There are hepatocytes, Kupffer cells, itos cells, and pit cells. They are specific hepatic cells, and also we have here endotheliosides, which form wall of sinusoid. They aren't specific, but they also are present in liver. And in sinusoid, we can see here blood circulation, blood formed elements are circulating, and they are going to the central vein of from sinusoid. And here we can see main liver cells. There are hepatocytes. Also, we have here itos cells, there are stellate macrophages or Kupffer cells, and also there are pit cells, main liver cells. And here we can see hepatocytes. There are endothelial cells, and it's itos cell, which is located in the Disse space, and it's Kupffer cell. And on this picture, we can see cross-section, transverse section of the sinusoid. Here we can see hepatocytes, its space of Disse, and its sinusoid. There are endothelial cells, and between them there is Kupffer cell, stellate macrophage, and continuous basal membrane is present here. And in space of Disse, between the hepatocytes, we can see triangular itos cells. And now let's go to the next question. It's blood supply of the liver. Liver receives blood from two sources. It receives blood from portal vein, and here we can see portal vein. It collects blood from organs of abdominal cavity, from intestines, for example, and it takes blood which contains different nutrients which are absorbed by intestines. And different toxins and harmful factors may be present there. So portal vein carries this blood to liver, and liver provides filtration and neutralization, purification of those blood, and it provides detoxification of different harmful substances. So all blood from abdominal cavity should be filtrated via liver. But liver also needs arterial blood, not only venous blood from portal vein. It also needs arterial blood for own needs for own nutrition, and also liver needs oxygen from arterial blood. So hepatic artery is the second source of the blood supply of the liver. It carries blood from aorta. It's the secondary branch of aorta, so it origins from pre-existing arteries, and here we can see hepatic artery which enters in the liver, and also it carries blood to the liver. So in liver we have two sources of blood, it's portal vein and hepatic arteries. And here we can see that inside the liver they follow each other, and also they are followed by branches of bile ducts. Uh, Portal vein and hepatic arteries branches they are going to the lobules, and bile ducts they follow them, but they carry bile from the lobules to the common bile duct. And here we can see blood circulation inside the hepatic lobule. In hepatic lobule, we have sinusoids which receive blood from two sources, from portal vein and hepatic artery, and their branches are located near the um, hepatic lobules as interlobular vein and interlobular arteries. So artery and vein, they are divided into smaller and smaller branches, and they achieve lobules. They are located between the lobules and the interlobular connective tissue as interlobular artery and interlobular vein. They are present here, and they provide blood supply, uh, and blood, venous and arterial blood goes to sinusoid. In sinusoid we have mixed blood, and this blood from sinusoid after the filtration goes to the central vein, and central vein collects venous blood and carries it away. So here we can see that branch of portal vein and branch of artery and also small bile duct, they form a bundle which contains three components. This bundle is called hepatic triad. It includes interlobular artery, interlobular vein, and interlobular bile duct. Here we can see hepatic artery, it's interlobular artery, branch of hepatic artery. It's vein, branch of the portal vein, it's interlobular vein, and also there is interlobular bile duct, which is located near the artery and vein. And those three um, vessels, two vessels and one duct, they usually form together hepatic triad, so they are located uh, near each other. And those hepatic triads, they are located near the angles of the hepatic lobule. Hepatic lobule is hexagonal in shape, so near the angles of this hexagon, there are interlobular artery, interlobular vein, and interlobular bile duct. It's called hepatic triad. Here we can see hepatic lobule, it's central vein, neighboring hepatic lobules, it's interlobular connective tissue, and near the ankles we have interlobular artery, interlobular vein, and interlobular bile duct. It's called triad. And triads are located near the ankles of the hepatic lobule. So each hepatic lobule it has neighboring artery, vein, and bile duct, and they receive blood from artery and vein, and they release bile to this bile duct. So here we can see blood circulation in the hepatic lobule. There are artery, vein, and its central vein. This artery is branch of the hepatic artery, and this vein is the branch of portal vein. So it's interlobular artery, interlobular vein, and blood, arterial and venous blood goes from those sources. And in sinusoids, it's mixed. So sinusoids, they contain mixed blood, and mixed blood goes from the periphery to the center, And after the filtration, after passing through the sinusoids of hepatic lobule, this blood becomes to be venous, and venous blood is collected by the central vein. So mixed blood goes through the sinusoids and goes to the central vein. Here we can see artery vein, there are sinusoids, and its central vein. And here we can see central vein. There are hepatocytes and sinusoids between them are present. Sinusoids, hepatocytes or hepatic plates, and sinusoids, they carry this blood to the central vein. Its artery, its portal branch of the portal vein, and mixed blood goes to the central vein. This localization of sinusoids, capillaries between vein and vein, it's called rete mirabile venosum. When capillary network is located between two vessels with the same name, between vein and vein. And also we have rete mirabile venosum in hypophysis when we have portal vein of hypophysis and hypophyseal vein. There are capillaries between two veins. And we have rete mirabile arteriosum in kidneys. Between two arterioles we have capillaries. There are two arterioles, and we will study this in the following topic. So here we can see sinusoids. They contain blood which goes here from two sources, from artery and vein. And this blood from artery, from vein goes to sinusoid. Sinusoid contains mixed blood, and this blood goes to the central vein, which collects all blood from the hepatic lobule. And here we can see the beginning of the central vein. There are hepatic plates composed of hepatocytes, and there are sinusoids which are opened here. So blood circulation of the liver from the periphery to the center of the hepatic lobule. And bile goes from the center to the periphery. So blood and bile, they are circulating in different directions. Blood from periphery to the center, and bile goes from the center to the periphery and goes to the bile ducts, which are located between the lobules. There are interlobular bile ducts. And here we can see hepatic plate composed of hepatocytes. There are sinusoids. Next, hepatic plates, its central vein, basic membranes. Also, Kupffer cells are present here, and blood goes from the periphery to the center. Blood is mixed here, and it passes through the hepatic plate and goes to the central vein. And as a result of it, it becomes to be venous. But this venous blood is filtrated. And central vein, it forms sublobular veins which excite from the lobules, and they form also network, and they form as a result hepatic vein. So hepatic vein collects all blood, all venous blood from the hepatic lobes from the liver. So central vein forms sublobular veins, and sublobular veins they form hepatic vein, which carries blood to the vena cava inferior. And let's look once again. So here we can see hepatic plate, endothelium of sinusoids, in other hepatic plates, its central vein, basic membrane, Kupffer cell, which is immune cell, and blood comes here. It goes from periphery to the center, mixed blood of sinusoid, and it carries different substances, different metabolites, different drugs, medicines, toxins, for example, everything what have been absorbed in the intestines. All those substances are going to the portal vein, which carries this blood to the liver. So this blood goes through the sinusoids, and it contains different substances. For example, something is coming here. This substance goes through the endothelium, through the space of Dissea to the hepatocyte. Here it undergoes detoxification or further metabolism, and after that it's released via bile surface to the bile canaliculus, and bile goes from the center to the periphery of the hepatic lobule. And here we can see movement of blood and bile in the hepatic lobule. And we can see that they have different directions, opposite directions of the movement. And here we can see hepatic lobule, it's classical hepatic lobule, and also we have some additional structural and functional units. Also, some scientists, they describe hepatic eosinophils. It's a rhomb-like region between two central veins and two triads. Here we can see hepatic eosinophils. And also, some scientists describe portal lobule triangle between three central veins which surrounds triad. So here we can see portal lobule. These triads, they are followed by a thin layer of connective tissue which is called portal tract, and triangle which surrounds this portal tract is called portal lobule. So here we can see alternative structural and functional units of liver, but usually we describe classical hepatic lobules, which is hexagonal in shape. And also the organ which is associated with liver is gallbladder. Here we can see gallbladder, and this bladder, it has structure which is similar to the wall of digestive tube. It has layered structure, and it has mucosa, its epithelium, simple columnar and lamina propria. It has muscularis, so some mucosa is absent here because it forms folds, and under the mucosa directly we have layer of smooth muscles. They mainly are oriented in oblique direction. And the outer layer is usually adventitia, or sometimes it's serosa according to the localization of gallbladder. When it's covered with peritoneum, it has serosa, and in places where it's not covered with peritoneum, it has adventitia. It's loose fibrous connective tissue. But in some textbooks, you can find that adventitia and particularly lamina propria, they are composed of dense irregular connective tissue. This dense connective tissue, it provides bigger density and it maintains the shape of the gallbladder, and it prevents the collapsing of gallbladder. Here we can see a structure of gallbladder. It's mucosa, it's muscularis, and adventitia. And bile ducts, they also have structure similar to the structure of gallbladder. Also, there is mucosa, muscularis, and adventitia. And muscularis disappears in smaller branches of bile ducts, and it's absent in small bile ducts. They have only epithelium, basal membrane, and thin layer of loose fibrous connective tissue. They are located as a part of triad inside the portal tracts near the angles of hepatic lobes. So, students, we have discussed structure of liver and bile ducts and gallbladder. So that's all. Thank you for your attention.